Hello everyone, I am Aniket Dattatreya Takle. Today we are discussed about DNA replication. So let's start. When we are studying about DNA replication, all of you know that what is meant by DNA. DNA is nothing but the group of molecules that is responsible for carrying and transmitting the hereditary materials or the genetic instructions from parents to offsprings. In that, DNA is known as deoxyribonucleic acid. It is an organic compound that has a unique molecular structure and it is found in all prokaryotic cells and eukaryotic cells. Next part is DNA types. There are three types of DNA. First one is aDNA, second one is bDNA, and third one is zDNA. In that aDNA, it is a right-handed double helix similar to the bDNA form. Dehydrated DNA takes an A form that protects the DNA during extreme conditions such as desiccation. Protein binding also removes the solvent from DNA and the DNA takes an A form. Second one is bDNA. In a bDNA, this is the most common DNA conformation and is a right-handed helix. The majority of DNA has a B-type conformation under normal physiological conditions. In the third type, that is zDNA. In that zDNA, it is a left-handed DNA where the double helix winds to the left in a zigzag pattern. It was discovered by Andres Wang and Alexander Rich. It is found ahead of the start site of a gene and hence is believed to play some role in gene regulation. Next point is who discovered DNA. DNA was first recognized and identified by the Swiss biologist Johannes Friedrich Miescher in 1869 during his research on white blood cells. In that, the double helix structure of DNA molecule was later discovered through the experimental data by James Watson and Francis Crick. Finally, it was proved that DNA is responsible for storing genetic information in living organisms. In that, DNA comprises a sugar phosphate backbone and the nucleotide bases which includes guanine, cytosine, adenine and thymine. Next point is DNA structure. In the DNA structure, the DNA structure can be thought of as a twisted ladder. This structure is described as a double helix as illustrated in the figure. It is a nucleic acid and all nucleic acids are made up of nucleotides. The DNA molecule is composed of units called as nucleotides and each nucleotide is composed of three different components such as sugar, phosphate groups and nitrogen bases. The basic building blocks of DNA are nucleotides which are composed of a sugar group, a phosphate group and a nitrogen base. In that the sugar and phosphate groups link the nucleotides together to form each strand of DNA. That means adenine, thymine, guanine and cytosine are four types of nitrogen bases. These four nitrogenous bases pair together in the following way, that is A adenine with thymine and cytosine with guanine. These base pairs are essential for the DNA's double helix structure which resembles a twisted ladder. Among the three components of DNA structure, sugar is the one which forms the backbone of the DNA molecule. It is also called deoxyribose. The nitrogenous bases of the opposite strands form hydrogen bonds forming a ladder-like structure. In that, the DNA molecule consists of four nitrogen bases, namely adenine, thymine, cytosine, guanine, which ultimately form the structure of a nucleotide. The adenine and guanine are purines, and the cytosine and thymine are pyrimidines. The two strands of DNA run in opposite directions. These strands are held together by the hydrogen bond that is present between the two complementary bases. That means the strands are helically twisted, where each strand forms a right-handed coil and 10 nucleotides make up a single turn. The pitch of Each helix is 3.4 nanometer, hence the distance between two consecutive base pairs, that is hydrogen bonded bases of the opposite strands, is 0.34 nanometer. In that, the DNA coils up forming chromosomes, and each chromosome has a single molecule of DNA in it. Overall, human beings have around 23 pairs of chromosomes in the nucleus of cells. DNA also plays an essential role in the process of cell division. Next point is Chargaff's rule. In that, Erwin Chargaff, a biochemist, discovered that the number of nitrogenous bases in the DNA was present in equal quantities. That means the amount of adenine is equal to the thymine, whereas the amount of cytosine is equal to the guanine. In other words, the DNA of any cell from any organism should have a 1:1 ratio of purine and pyrimidine bases. So main part is DNA replication. In the DNA replication is an important process that occurs during cell division. It is also known as semi-conservative replication, during which DNA makes a copy of itself. There are three steps of DNA replication. First one is initiation, second one is elongation, and third one is termination. In, the, in that initiation step, the replication of DNA begins at a point known as the origin of replication. That means the two DNA strands are separated by the DNA helicase. This forms the replication fork. Second step is elongation. In the elongation step, DNA polymerase third reads the nucleotides on the template strand and makes a new strand by adding complementary nucleotides one after the other. For example, if it reads an adenine on the template strand, it will add a thymine on the complementary strand. While adding nucleotides to the lagging strand, gaps are formed between the strands. That means these gaps are known as the Okazaki fragments. These gaps are nicks are sealed by ligase. Third step is termination. In the termination step, the termination sequence present opposite to the origin of replication terminates the replication process. That means the TUS protein, that means terminus utilization substance, binds to terminator sequence and halts DNA polymerase movement. It includes termination. These are all about DNA replication. Thank you.